Main arena of various cell activities. Cytoplasm. Viruses 0.0 to 0.2 micrometer. PPLO 0.1 micrometer. Eukaryotic cells 10 to 20 micrometer. Bacterium 2 to 3 micrometer. In prokaryotes, mitochondria absent. Krebs cycle occur over cytoplasm. Prokaryotes in plant cell occurs cell wall. Membrane extension in cytoplasm. Cyanobacteria. Chromatophore pigment. Membranous extension of in cytoplasm is chromatophores. Mitochondria in bacteria. Mesosome. Mitotic division absent. E. coli. Layers around cell wall of bacteria. Glycocalyx. Capsule. Immune to bacteria. Smallest blister-like fibers sprouting out cell. Attach host tissue. Fimbriae. Ribosomes. Eukaryote 80s. Prokaryote 70s. 20 to 30 nanometer in diameter. 2 subunits. Mesosomes. Only seen in prokaryotes. Extent of plasma membrane into cell helps in cell wall formation. Polysomes. Group of ribosomes. Prokaryotic ribosome 70s. Smooth endoplasmic reticulum. Lipid synthesis. Gram-negative bacteria. E. coli. Bacteria move through flagella. Gram-negative and gram-positive main difference: cell wall and envelope. Cell envelope. Plasmid replicate independently. Protoplast cell with or without cell wall. Eukaryotes not have peptidoglycan in cell wall. Prokaryotes and eukaryotes. Cell membrane. Non-membrane organelles: centrioles, ribosome, nucleolus. Plasma membrane. Proteins embedded in phospholipid bilayer. Protein, lipid, carbohydrate. 70s. Bacteria. बैक्टीरिया ये क्या है? Nostoc chloroplast. Ribosome composed of RNA and protein. Plant cell, difference animal cell wall and chloroplast. Plant cell, large vacuole and rigid cell wall. Na plus and K plus pump. Active transport. Transmembrane protein in lipid bilayer. Act as channel proteins. Transport without the help of protein in biomembrane. Simple diffusion. Electron microscope for detailed study cell. 1950s. Active transport characteristics: highly selective, uphill transport in saturation, not sensitive to inhibitors. Active transport acquires special membrane protein, highly selective, requires ATP energy. Ingestion of solid or liquid substance into cell is endocytosis. Ingestion of a solid or liquid substances into cell is endocytosis. Constituent of cell membrane: glycolipid, phospholipid, cholesterol. Not constitute of cell membrane: protein. Na plus K plus pump, Na plus oral, Na plus out and K plus in. Most abundant lipid in cell membrane is phospholipid. Endocytosis engulfing and internalizes material with its membrane. Cell membrane are made of phospholipid and proteins. Animal cell not have plasmodesmata. Between adjacent cell is a effective transport pathway. Plasmodesmata. Strength and rigidity of cell wall. Cellulose. Plant cell not surrounded by cell wall. Gamete cell. Strength and rigidity of cell wall. Cellulose. Plant cell not surrounded by cell wall. Gamete cell. Middle lamella mainly composed of calcium pectate. Cell connects by plasma data forms semplast. DNA contains mitochondria, plastids, nucleus, chloroplast. ATP required for active transport. Eukaryotic cell, difference prokaryotic cell, true nucleus. Membrane most abundant in cell is ER membrane, endoplasmic membrane. Part of endomembrane system, eukaryotic cell, Golgi vacuole, Golgi body, vacuole, endoplasmic reticulum, lysosome. Correct fluid mosaic model. Lipid can rarely flip-flop, protein cannot. Oxidative organelle, peroxisomes. Hydrolytic enzyme bag, lysosomes. Photorespiration, peroxisome. Interconnected flat sacs of ER, cristae. Chloroplast, thylakoid. Origin of lysosomes, endoplasmic reticulum, Golgi complex, lysosomes. Interconnected membrane network cell compose veins, vesicles, cristae, tubules, ER. Figure Golgi body. Endoplasmic reticular detoxification. Spherosomes, lipid synthesis. Packaging and secretion, Golgi apparatus. Golgi apparatus not contain ribosomes. Ribosomes in mitochondria are ER. Golgi complex major role post translational modification of protein and glycosylation lipids. Intercellular digestion, lysosomes. Respiratory enzyme, mitochondria. Hydrolysis vacuoles, food, lysosomes. Vacuoles, high concentration of sap than cytoplasm. Vacuoles, topoplast, membrane bound, contains water and counter substances. Two membrane envelope, mitochondria, nucleus, chloroplast. Mitochondrial diagram and description and differences with and similarities with chloroplast देख लेना एक बार, बहुत इंपॉर्टेंट है. देख लेना एक बार, नहीं देखना ही देखना है. एनर्जी रिलीज reaction in cell occurs mitochondria. Both plants and animals, mitochondria. Projecting inner surface of cristae and inner mitochondria membrane, oxysome, F0 and F1 particles. Mitochondria semi-autonomous, DNA plus RNA plus ribosomes. Chloroplast in higher plants, lens shape. Lipid contains stored food material, Elioplast, Uroplast, Amyloplast. Plastid not contain stored food material, Keratinoids, Chromoplast. Thylakoid, flat membrane sacs in chloroplast. Leucoplast, no pigment. Difference between mitochondria and chloroplast, thylakoid membrane, ये सब NCERT से पढ़ना है, ठीक है. Mitochondria, enzyme of electron transfer chain are embedded in inner membrane. Quantizome photosystem grana. Common chloroplast, chromoplast, leucoplast divided by bringing fission. Thylakoid inner side chloroplast, absent in animal cell, chloroplast and plastids. Not true organelles, ribosomes. Largest subunit of prokaryote ribosomes, 50S. 50S ribosome subunit composed 23S RNA and 5S RNA and plus 30 type of proteins. Ribosomes discovered in animal cell by Pallade. Cytoskeleton protein proteinaceous filament. Length of mitochondria 1 to 4 micrometer long. Width of mitochondria in diameter is 0.2 to 10 micrometer. Average is 0.5 micrometer. TCA enzyme mostly mitochondrial matrix. Exonyma of cilia plus 9+2 array. Stroma chloroplast colorless fluid inside enzyme photosynthesis chloroplast color pigment. Exonymal arrangement 9 pairs of doublets and 1 central pair of singlets. सीलिया का पैराग्राफ देखो, बहुत इंपॉर्टेंट है. टोटल माइक्रोट्यूब्यूल्स एंड फ्लैजिला और सीलिया 20. Microtubules are made up of tubulins. Centrosome absent in cells of higher plants. Proteins required inside nucleus formed in cytoplasm. Very long and very short acrocentric. One short and one longer submetacentric. लॉन्ग क्यों? क्वेश्चन लंबा है, इसको ऐसे याद रखेंगे. क्वेश्चन लंबा है तो क्वेश्चन चालू होता है क्यू से. तो क्यू आर्म इज लॉन्गेस्ट एंड पी शॉर्टेस्ट. टर्मिनल सेंट्रोमियर telocentric. Chromatin coined by Fleming. Nucleolus contains chromatin portion. Structure element of chromatin nucleosome. Nucleolus set chromosome. Shape of chloroplast in higher plants discoid. Solenoid coiling of DNA packaging chromatin fiber with 30 nanometer diameter. Chromatin chemically made up of nucleic acid, histones and non-histones protein. rRNA synthesize nucleolus. Enucleated plant cell, sieve tube cell. Transport of RNA and protein through nucleus to cytoplasm. Nuclear pore complex. Porins are located in mitochondria complex in their outer membrane. Transcription occurs of euchromatin. Flagella occurs in prokaryote and eukaryote both. 9+2 array, cilia flagella both. Cell division. RNA and protein synthesized in G1 phase and G2 phase. Mitotic cell cycle G1 S, G2 M, G1. Drug colchicine inhibitor effect on M. Yeast complete cell cycle in 90 minutes. GO. Cell metabolic continuous in G0. Cell growth occurs in G0. Biocatalytic help exist G0. G1 phase, interval between mitotic and initiation action of DNA replication. S phase, replication of DNA. G2 phase, protein synthesis. G0 phase, question stage. Centriole and centrosome replication. S phase, interphase. Chromosome number remains same in S phase and DNA replication. Occurs in interphase of eukaryotic cell division. Increase ATP, DNA, RNA synthesis. Not occurs in interphase of eukaryotic cell division. Reduction in cell size. G1 stage of interphase of cell cycle shows active synthesis of RNA, divide active protein synthesis. In mitosis, chromosome duplicates occurs during interphase. 2C becomes 4C DNA in G2 में है. Mitotic divisions needed for single cell to make 1024 cells stem. 256 daughter cells. Number of mitotic division 8. Syntheticum karyokinesis not followed by cytokinesis. Mitosis somatic cell. Meiosis germ cell. Equal distribution of chromosomes, splitting of centromeres. During mitosis, endoplasmic reticulum and nucleolus begin disappear. Slit prophase. Smaller cell have large surface area volume ratio. Anaphase stage of mitosis, centrosomes split, chromatid separate, chromatid move to opposite poles. Shape of chromosome best observed during metaphase. Smallest disc-shaped structure at structure of centromere is kinetochore. Liquid in coconut because karyokinesis is not followed by cytokinesis. Stage of cell division, chromosomes are most condensed. Metaphase. Keep cell in metaphase stage of mitosis is in blood culture technique colchicine. Spindle fiber formed of tubulin protein. Colchicine arrests stage of mitosis metaphase. Colchicine interfere with cell division because non-formation of spindle. Significance of mitosis cell repair. Onion root tip during metaphase 34. Example of mitogen cytokinin. Structures, number of shape and of chromosome observed. Metaphase 1. Meiosis. Displacement of chiasmata occurs in dikinesis. Separation of two basic sets chromosomes. In meiosis, synapses occurs during prophase. In meiosis, chromosomes now become half of the plant cell. In meiosis, first cell division is reductional and second equational. Enzyme recombinase meiosis penchidin. Pair synapses bivalent formation. Zygotin. Arrange synapses crossing over termination of chiasmata disappear nucleolus. Anaphase chromosomes move to opposite poles. Anaphase 1 homologous chromosomes separate. Anaphase 2 centromere splits. Homologous chromosome pairs up only meiosis 1. Meiosis does not occur in haploid cell. Reorganization recombination meiosis. Synaptomer complex dissolve diplotin. Chiasmata was seen in diplotin. डिप्लोटीन हो सकता है. Synaptomer complex dissolve in diplotin. Chiasmata was seen in diplotin. Non-sister chromatid exchange segment during panchitin. Recombination involve crossing over. Homologous chromosomes separate during anaphase 1. Crossing over take place in prophase 1. Initiation of spindle fiber zygotin. Synthesis of RNA in protein G1. Action of endonuclease panchitin. Movement of chromatids towards opposite poles anaphase 2. Homologous chromosomes separate while chromatin at their centromere anaphase 1. Synaptonemal complex site of chromosome align and recombination. Homologous gene appears at anaphase 1. Leptotin visible of chromosome. Thin log thread like. Leptotin. Zygotin synapses. Panchitin crossing over and recombination. Dikinesis terminalization of chiasmata. Bivalent metaphase 1 consists of 4 chromatids and 2 centromeres. Not character of meiosis. 2 stage of DNA replication. One first before meiosis 1 and second before meiosis 2. Meiosis best seen in microsporangium. Dikinesis equatorial plate nucleolus disappear. Chromatid chromosomes clearly visible in meiosis panchitin. Terminalization occurs during dikinesis. Microtubules from opposite poles of spindle gets attached to sister chromatids during metaphase 2. Longitudinally divided into two identical parts, sister chromatids. Plane of cell wall formation in the dividing cell is determined by microtubules. Meiosis 1, number of chromosome is halved. G2, production of energy required for spindle formation. Prometaphase, fusion of microtubules to form spindle apparatus. Anaphase, contraction of tubulin protein. Panchitin, recombination of genetic material. Unique to mitosis but not occur in meiosis. Homologous chromosomes, some behaves independently. Mitosis, daughter and parent cells have same chromosome number. Meiosis, synapses of homologous chromosomes. G2 phase, production of energy required for spindle formation. Prometaphase, fusion microtubule to form spindle apparatus. Anaphase, contraction of tubulin protein. Panchitin, recombination of genetic material. Exchange of segment between parental and maternal chromosomes, crossing over. Spindle fibers that connects the centromere to respective poles, chromosomal fiber. Biomolecules, 50 gram of living tissue, 35 to 45 gram H2O, aromatic amino acid, tyrosine and tryptophan, not hydrolyzed further, ribose. Fats, glycerol in trihydroxypropane. Fatty acid, palmitic acid. Lectin, a component of cell membrane. Lectin, phosphorylated glyceraldehyde formed in cell. Palmitic acid, 16 carbon, CH3, CH2, 14, COOH. Adenylic acid is carbon based sugar phosphate group. Alanine amino acid, NH2, CH3, COOH or H. Amino acids are substituted methanes. Glycerol, trihydroxypropane. Lysine, basic amino acid. Lectin, structural lipid of cell membrane, phospholipid. Essential amino acid is valine. Not essential amino acid is tyrosine. Element with protoplasm and proteinaceous material